To read millions of DNA sequences simultaneously, high-throughput sequencing historically relies on heavy optical machinery. We expect systems requiring lasers, cameras, and complex flow cells. Ion Torrent abandons the camera. Instead of detecting light, it uses a semiconductor pH sensor, a solid-state silicon chip that directly detects chemical changes. Optical systems require expensive, modified nucleotides. Each DNA base must be attached to a fluorescent tag so that a camera can actually see it as it is incorporated. By replacing the camera with a microchip, the sequencing reaction can run on unmodified, standard DNA nucleotides. The hardware is designed to detect a naturally occurring chemical byproduct of DNA synthesis itself. That byproduct is a single unbound hydrogen ion, a proton. Translating these chemical byproducts directly into an electrical voltage eliminates the need for complex reagents and lowers the cost of a sequencing run. That measuring free-floating protons introduces entirely different physical limits. The process starts at the sub-nanometer scale. The sequencing reaction uses standard DNA polymerase and unmodified nucleotides. When the growing DNA strand incorporates a new base, the hydroxyl group on its 3' end attacks the alpha phosphate of the incoming nucleotide. Forming this new phosphodiester bond forces the cleavage and release of a pyrophosphate molecule. Alongside that pyrophosphate, a single hydrogen atom is cleaved from the hydroxyl group and ejected directly into the surrounding solution as a free proton. Free protons increase the acidity of a liquid. Every time a base is added, the local microscopic environment becomes infinitesimally more acidic. This establishes a strict mathematical correlation. The incorporation of exactly one base guarantees the release of exactly one hydrogen ion. The act of building DNA inherently generates a reliable acidic footprint. No lasers or fluorescent tags are required. The chemistry provides its own trigger. To capture that tiny acidic footprint, the reaction is isolated inside a microscopic femtoliter volume well built directly onto the silicon chip. Inside this well sits a single spherical bead coated with approximately 1 million identical clonal copies of a single DNA fragment. When a matching nucleotide is introduced into the well, 1 million polymerases incorporated across all those DNA copies simultaneously. At the absolute bottom of this well lies the sensory hardware, an ion-sensitive field effect transistor, or ISFET. The simultaneous release of 1 million hydrogen ions causes a sharp, highly localized drop in the pH of the fluid trapped inside the well. The ISFET detects this rapid concentration change and translates the drop in pH directly into a proportional electrical voltage spike. Every single well operates as a microscopic battery. The biological act of DNA synthesis generates the electrical charge that powers the machine's data output. But there is an operational problem. Every base pair, whether A, C, T, or G, releases the exact same hydrogen ion. The voltage spike tells us a base was added, but it doesn't tell us which one. To identify the bases, the chip relies purely on sequential timing. We wash one type of nucleotide over the chip at a time. In cycle 1, the well is flooded with only A nucleotides. If the DNA template requires an A, protons are released and the ISFT records a voltage spike in the A column. If the template does not require an A, the polymerases do nothing. No protons are released and the voltage line remains completely flat. The A nucleotides are washed away, and the cycle repeats with C, then G, and finally T. If there is a spike during the C wash, the sequence records a C. Avoiding fluorescent tagging and using standard nucleotides drastically simplifies the reagent supply chain, which keeps the financial cost of a sequencing run low. Scaling this up, a single Ion 540 chip houses over 80 million of these microscopic wells running the sequence in parallel. Running an Ion 540 chip to completion takes between 4 to 8 hours and generates a maximum of roughly 15 gigabases of sequencing data. For context, an optical system like the Illumina NovaSeq can produce upwards of 1,000 gigabases in a single run. A whole human genome sequence requires 30 to 50 gigabases of data just to establish proper coverage. While a single Ion Torrent chip is inexpensive to buy, its low throughput means you would need multiple chips just to finish one genome. For large-scale projects, the price of these individual chips adds up quickly, making semiconductor sequencing economically inefficient for massive genomic projects compared to high-volume optical platforms. While the platform boasts an overall accuracy rate of 99%, it faces a specific physical limitation. It struggles to accurately read homopolymers, runs of multiple identical bases in a row. Imagine a DNA template requiring 8 A bases. Introducing all A nucleotides at once makes polymerases incorporate all 8 simultaneously. In theory, this yields a perfectly linear pH drop of 8 units. In reality, the pH drop curves off, flattening around 5 units. This nonlinear signal happens for a few physical reasons. First, as a massive wave of hydrogen ions floods the well, many simply diffuse out of the open top into the surrounding fluid before the sensor at the bottom can register them. Second, the fluid in the well possesses inherent buffering capacity that resists extreme shifts in pH, and the pyrophosphatase enzymes responsible for clearing out chemical waste quickly become saturated and inefficient. Finally, the ISFET sensor itself hits the limits of its dynamic range when overwhelmed by massive, sudden swings in acidity. The physical dynamics of fluid buffering and ion diffusion ultimately break down the clean one-to-one mathematical correlation between base incorporation and voltage output during long sequences. When physical saturation occurs, it manifests as a critical bioinformatics error. A true sequence of 8 A's is recorded as 6 A's. Because the hardware registers a 6-unit pH drop, the software deletes 2 bases, creating an insertion or deletion error known as an indel. In coding regions of a genome, an indel is catastrophic. Missing a single base pair causes a frame shift mutation, which shifts the entire reading frame of the sequence and renders the translated proteins completely non-functional in analysis. For homopolymers under 5 bases long, the error rate remains under 1%, but once a sequence stretches past 8 identical consecutive bases, the error rate spikes to anywhere from 10 to 50%. Signal ambiguities and the accumulation of indel errors compound over successive sequencing cycles. To maintain viable accuracy, Ion Torrent is physically forced to cap its functional read lengths at roughly 400 to 600 base pairs. Ion Torrent bypasses the massive data ceilings and extended read lengths of optical sequencing to achieve the speed, simplicity, and low cost of electrochemistry. It is a highly efficient tool for targeted diagnostics, yet it remains forever bounded by the unyielding physical limits of a single free proton.